Next up, we have Dr. Ray Takagiku, President and CEO of Bexion Pharmaceuticals. Ray, take it away.
Great. Thank you, Katie. So as Katie said, my name's Ray Takagiku. I'm founder and CEO of Bexion Pharmaceuticals, and I'm very happy to be able to talk to you today about our lead compound, BxQ350. So we are a clinical stage oncology company, and BxQ350, our lead compound, is composed of a lysosomal protein called Saposin C. And Saposin C is an endogenous human lysosomal protein. It's a small protein, but it has some pretty remarkable properties which are particularly important to the treatment of cancer. This protein is a regulator protein, and so one of the things that it does is activate catalysis of ceramide, which is the mechanism by which it causes cell death. It has a couple of other unique attributes which lend itself to being a cancer therapeutic, and one of those is that it's only active in acidic microenvironments since it is a lysosomal protein, and as all solid tumors have an acidic environment, it is only active in those, around those tumor tissues and not around healthy tissues. The other property that Saposin C has is that it, in fact, needs a negatively charged lipid in order to be active. And that negatively charged lipid is called phosphatidylserine. And it so happens that all solid tumors ubiquitously express phosphatidylserine. They do that because phosphatidylserine is immunosuppressive. And so as we intravenously administer BxQ350, the Saposin in BxQ350 is attracted to those phosphatidylserines because it's in an acidic environment. And so we see some remarkable tumor targeting in our preclinical models with labeled compound. We even see it to metastasized tumors. So the important thing to know about how BxQ350 works against tumors is there are two sphingolipids which play counter roles in cell homeostasis. Ceramide, elevation of ceramide leads to cell death, and generation of a lipid that's generated from ceramide called sphingosine 1-phosphate, or S1P, leads to cell survival. So there's this interplay of cancer cells in which sphingosine 1-phosphate is elevated. And what happens when we administer BxQ350 is we are through those natural mechanisms by which Saposin C activates these sphingolipid enzymes, we generate ceramide. And so that's the middle slide. It shows you that there are at least 3 ways in which ceramide is being generated in tumor cells. And on the right, you'll see some graphs. The top graph is a preclinical model of glioblastoma, and you'll see after treatment with BxQ350, there is this elevation of ceramide. We also happen to see it in our phase 1 patient population. So this is a GBM patient, a patient with glioblastoma, and after cycle 1 or day 29, ceramide also was elevated. So consistent with this increase in ceramide, BxQ350 causes cell death by a variety of different mechanisms. You see necrosis, apoptosis. I want to call your attention to the electron micrographs on the left. So the top 2 micrographs are from GBM neurospheres. So that's, again, a Petri dish type experiment. And very surprisingly, after BxQ treatment, there was this observation of autophagic vacuoles. Now, fortunately, from our phase 1 study, we were able to obtain some biopsies from GBM patients. And when those biopsies were examined under the electron microscope, we saw similar sort of autophagic vacuoles. So that was a very interesting finding, and definitely we're following up with further research on that observation. I want to share with you a couple of case studies from our phase 1 program. because of that tumor targeting effect that I described, we ended up doing both an adult and pediatric program, over 1,100 doses of BxQ, almost 100 patients, a variety of different tumor types. And what we saw was a very safe profile from that phase 1 program. So one of the things that we're looking forward to is using BxQ along with other chemotherapies, standard of care across different cancers, to improve patient outcomes. I did want to show you a couple of these case studies, and in this case, this is a 64-year-old male with recurrent GBM. You see that GBM in the leftmost MRI, and about a year after being on BxQ, there was this secondary area of enhancement. Surgery was performed, and basically that tissue was found to be necrotic. And you can look at the most recent MRI we have of that patient, and that was on May of 2020. This particular patient has been on BxQ for over 5 years and is almost 5 years and is doing very well. This is a colorectal cancer patient. This is a stage 4 female who came into our study, was being recommended a hospice, but fortunately agreed to come on BxQ. You can see her PET scan there on the left prior to coming on to our study, and you can see her PET scan a year after being on our study. So we have elected to pursue in phase 2 trials a pediatric program in a rare pediatric disease called DIPG-DMG, and we're also looking forward to opening up a phase 2 program here in colorectal cancer, and we'll be eventually looking forward to opening up a phase 2 in glioblastoma. So I'm going to end up with this milestone slide, and it's a busy slide, but I want to point your attention to a couple things. One of them is the National Cancer Institutes on the top left. Bexion is privileged to be one of the highest private company grant recipients from the NCI. In fact, we received a bridge award, which was vetted by Harold Varmus. So at the time, it was quite a nice validation of our approach to treating cancer. On the upper middle, you'll see FDA designations for pediatric rare disease designation, as well as orphan drug designation. So Bexion does have those, and those are programs intended to help us move forward from a regulatory front. Below the years, you'll see that we are either in phase 2 or, as I said, preparing to go into phase 2 for multiple clinical trials. Just below that, you'll see that we've got an extensive patent portfolio. And I did mention that Saposin C is this lysosomal protein, and on the cover side, you saw that we are a company focused on the lysosome. And so this approach in treating cancer through lysosomes actually has implications in other diseases, and different diseases, including peripheral neuropathy, antiviral, and Parkinson's. So we're excited about the programs we have moving in those areas as well. We're looking forward to a potential exit in 2023, and that's what our Open Series C is intended to help get us to. There are some comps on this slide.
Ray?
Yes.
Thank you so much for presenting everything about Bexion Pharmaceuticals, and I want to get on to the Q&A because we have lots of great questions as well.
Sure.
All right. I'm going to start with one of my own. What I heard from you is that Bexion is a systemic chemotherapy drug, but has limited toxicity based on the mechanism of action. So you presented some data, I know, on GI cancers as well as brain tumors, but I'm guessing it could have an impact on metastatic cancer as well. Is that true?
In fact, our phase 2 program that we're going into for colorectal is for metastatic colorectal cancer. So yes, because the drug is administered intravenously and because this drug seeks the ubiquitous tumor target of phosphatidylserine, we saw in preclinical models the ability of this compound to reach those metastatic tumors. So we're very hopeful that that will also be true. And we did see evidence of that in our phase 1 program as well, that we'll be able to treat those metastatic cancers.
Great. And talk a little bit more. I want to give you a minute to talk about a little bit more of the application of this therapy to other potential diseases and indications, because you threw that in at the very end. We need to hear more about that.
Yeah, one of the surprising findings that we had from our phase 1 program is we had some patients who had chemotherapy-induced peripheral neuropathy. So these were end-stage patients in our phase 1 program, patients that had gone through a variety of different chemotherapies. We had an average of 7 prior therapies. So we took a deeper dive at one of our sites at Ohio State at a cohort of patients who did come in with chemotherapy-induced peripheral neuropathy. And in fact, even one patient that came in with diabetic neuropathy. And at the end of the day, although it was anecdotal data because we were not collecting it in our cancer phase 1 study, what we saw were that 5 out of 6 patients who had long-term peripheral neuropathy saw relief as a result of being on BxQ. And it turns out that there are profound scientific reasons why that could be expected. And so, in fact, in our colorectal cancer trial, that is one of the secondary endpoints that we'll be looking for is actually prevention of chemotherapy-induced peripheral neuropathy.
Wow, that's very exciting. Super exciting. A lot of this takes me back to my medical school days, I have to say. We have a great question from our audience here about potential side effects. So if a patient were to become acidotic, say from an infectious or cardiac event, and they were, in the midst of being treated with your chemotherapeutic agent, would that be a concern? And is there any way to reverse the effect of BxQ?
Yeah, that's a great question. We did not have that kind of an event happen in our phase 1 patient population. So I wouldn't actually know how to answer that question except to say that at least in our animal studies, in all of our studies so far, we've not seen off-target effects.
Great. All right. Well, Ray, thank you again for being here and sharing Bexion Pharmaceuticals with us. We really appreciate it.